altogether, but not simultaneous. It will be little break and then we will start. And don't worry about your evaluation, okay? Because that is a very simple.
And I think beyond this 5-year program, if you have any questions, anything that you would want to discuss further, always feel free to email.
Yes, you have the email. That is the best way actually, because you document your question.
Yeah.
And he will also get time to respond.
Right.
Otherwise, sometimes you ask a hypothetical question and we try to give hypothetical answer, which is not correct.
Okay.
So that will be the best way.
Yeah.
That you have the email of both of us.
Yeah.
You can drop your query.
Yeah.
You plan your query what you want to do.
But have patience actually, because we get hundreds of emails every day. So yeah, but we will try to respond for sure. So yeah, today is the last day. We are meeting for the last time. So far as this program is concerned, hopefully we will meet again. But if you have any questions, just email, and of course we will try our best to respond.
Okay, so please carry on.
Yeah. Okay, so yeah, this would be rather a short session. So RNA-seq data analysis. So yeah, how many of you have done analysis of RNA-seq data? None? Oh, okay. So what kind of data are RNA-seq data? Transcriptomic data. And transcriptomic data, you understand, I think we discussed it. It is one of the omics data. DNA makes RNA and you get RNA transcripts. That data is transcriptomic data, okay? So RNA sequencing or RNA-seq technology is relatively recent technology. Before that, microarray was used, microarray technology, but it was soon replaced by maybe 10-15 years or so. So when I say soon, actually, so I'm thinking about that time scale when technologies are replaced. So it got replaced by RNA-seq because this technology is really very powerful and very useful. With RNA-seq technology, you can perform unbiased analysis of transcriptomic data, which means that you are not biased by, for example, what are known. So in microarray, you have the probes for the known genes, right? So you are constrained by the knowns. In RNA-seq, actually, you consider all transcripts that are arising from some experiment. That could be some condition under which an organism has been subjected. So you will collect all RNA transcript data, and then you would want to quantify the abundance of RNA transcripts. And some of the transcripts could be arising from genes that are not known. So you may identify some new genes. So gene discovery that we discussed. Okay. So gene discovery through RNA-seq, or you can find novel isoforms. Isoform, you understand? Isoform. What are isoforms? I'm sorry? Similar? No, not similar actually. Isoform. So,
variations or versions?
oh, variants. Oh no. So here actually, I am thinking about alternative splicing. So alternative splicing, you understand? What is alternative splicing? Yeah.
Sometimes one exam may skip.
Yeah. Right. So, which is actually a gene may give rise to multiple proteins through a process called alternative splicing. So where an exon, for example, sometimes it can act as coding, sometimes it can act as non-coding. So when it is acting as non-coding, it will get spliced out. Okay. So, and because of this, isoforms can be generated. Okay, splice isoforms. So RNA-seq can help in identifying the isoforms. So you can get the data, and there are computational tools that can resolve the isoforms that are there in the data. Okay, so let's keep going with these slides. They are not many, so I want to just appreciate what this technology is. Okay, so this is the workflow, and we will go one by one. So there might be a biological question. For example, I got a disease, and I just want to know which gene is responsible for my disease. Okay, so that is a biological question. And then I will collaborate with a wet lab biologist. I will say that let's design an experiment. Okay, so next step, experimental design. So I will say let's design an experiment. Collect RNA from me, but collect in triplets. So you understand replicates? Okay. So we want to understand biological variability. So that's why we want to get data in multiples of samples actually, representing different times could be, or different some different conditions or whatever you say from me. So at different times, or maybe the same region, but get it in triplets. So three times you get 3 samples, and then 3 samples from healthy individuals. Okay. So that is experimental design. So you are thinking about how many samples you want to get. So maybe 6 samples actually. So that's experimental design. And typically, if it is very expensive, then 3, but can be more. Can as many as possible, maybe 6 samples or even 10 samples if it's cheaper, because that gives more statistical power. Okay. Okay, and once the experiment is designed, then isolate RNA. Okay, so RNA will be isolated from me as well as a healthy individual or healthy individuals, multiple healthy individuals. Okay, so then here are the steps for RNA-seq. Once RNAs are extracted, so RNA library is made. So I will show you this again in a moment. And then the RNA library is subjected to sequencing, next generation sequencing. So this can sequence multiple of the RNA reads, and these reads are short fragments from RNA transcripts. Okay, so it can sequence millions to billions of these short sequences arising from RNA transcripts. Okay. So once these sequences are obtained, they are mapped back to the genome, and then quantify the abundance of reads for each of the genes in the genome, right? So when they are mapped onto the genome, you know that how many reads map to this gene, how many map to this one, and so on. Okay. So you will do this for healthy first, and then you will do this for me, actually, a sick individual, some disease. Okay, and then compare. So you want to identify which genes have difference in the abundance of reads for healthy and me. Okay, between healthy and me. So which have the differences. So for example, a gene may have RNA abundance of say 100 in healthy, and in me 1000. Okay, So how many fold more in me? 10-fold more in me. So it means that that particular gene is producing more RNA transcripts in me. Okay, and the disease that I am having may be arising from that. So again, that's a hypothesis, because that is indicative of something not right, because in healthy it is 100, in me it is 1000. Okay, so this is what RNA-seq helps in inferring, basically quantifying the abundance of RNA, so RNA transcripts in terms of the reads that are generated from this technology. Okay, so reads are mapped onto the genome, and then the expression level is quantified. So more is the abundance of RNA transcript, more is the gene expression. Okay, less abundance, less expression. So expression, when a gene expresses, it makes RNA transcripts. So more expressing, it will produce more RNA transcripts. Okay, very good. So this will result in hypothesis testing. So you have found that there is a difference in RNA abundance in a certain gene between healthy individual and me. But you would want to assess whether this difference is statistically significant or not. Is it arising because of a biological reason, or just by random biological variability? Okay, so you want to assess the statistical significance of this difference. So that's hypothesis testing. Okay, and you may also want to perform exploratory statistics. So perform clustering analysis. For example, principal component analysis of the samples. So 3 samples from me and 3 from healthy individuals. Okay, so perform PCA. You would expect that my samples, they will group together and will show as a group in the PCA plot, and those from healthy individuals, they will group together far from me. Okay, so you'd expect that, and also once you have obtained the genes that are expressing, or just the abundance of RNA transcripts in terms of the reads, counts of reads that mapped onto those genes, you can perform clustering of the genes in terms of their gene expression patterns. So basically in terms of the abundance of the RNA transcripts arising from them. Okay, so if 2 genes are expressing similarly, they would have similar pattern of transcript abundance, right? So they should group together. Okay, and then classification. So you can use machine learning algorithms, and you learned about machine learning yesterday, right? So you have the data coming from, say, different cancer patients. Some have breast cancer, liver cancer, lung cancer, and so on, and also healthy individuals, right? So you have got the RNA-seq data from them, and you can train a machine learning model on this data. Okay, so machine learning model will learn these differential patterns, representing differential patterns of transcript abundance, representing different cancers. Okay, so if there are 10 cancers, machine learning will learn these different patterns, 10 patterns arising from the 10 cancers, and then given new samples, it will predict which cancer is represented in which sample. Okay, so that is called classification. So it will classify the samples by cancer types. Clear? Okay, so and then biological confirmation. So if you have identified a gene that is important because it has a differential abundance, you would want to do some wet lab. So design experiment or assay to confirm the function of that gene. in the context of what you are interested in. Okay. Okay. So this will give you new novel biological insights, and it can also be used in diagnostics and therapeutics. So diagnostics, I just mentioned that you want to detect cancer. So classification can help, right? So there is a sample and maybe stage 1 cancer. So machine learning will identify that sample as stage 1 cancer. So great. Maybe biopsy is not able to, but machine learning model is able to. There could be some therapeutics to address this problem, right? So and the next is actually therapeutics. So RNA-seq can, so this is the workflow. So starting with the RNA. So starting with the biological question and then design and then so on. Okay, and you come to the end here. Okay. Okay, so RNA-seq data are provided in FASTQ format. So there are files and these are very large files. I have actually the workflow that one of my PhD students, he provided to me, so that I give that to you, and I think you have got the PowerPoint for that. We may not get time to do this, it takes time. But you have the PowerPoint, and I think I refined the PowerPoint, so I can send to you, and if there is a group email, you can forward them. Okay. So I did some refinement because he did not provide the accession numbers of all the samples, so now I have included them. So you can try that later on. And if you are a biology student, once in a while you may actually have to do such analysis because it's an era of big data, and these are the big data, RNA-seq data or DNA sequencing data. These are the big data. So it is possible that you may encounter at some point of time these data and you would want to analyze. So make sure that you have saved the RNA-seq pipeline that we have provided to you, right? So you will try, and if you have difficulty, then let us know. I will ask my student to help with that. Okay. So when I was doing, I was getting some server error, so, and which we have been actually with Galaxy, and he told me that just keep trying. Maybe in the third attempt or fourth attempt it will work, but it will work for sure. Okay. So his experience. So I was also wondering that why I am not able to get the answer, why I am getting the server error. So he said that yeah, it happens, and it is likely that it happened to him also, and he suggested that we should do it at different times. Maybe in the third attempt or fourth attempt it will work, because he has done it. All those samples are okay. He got the results, so you will also be able to. Okay. Okay. So here is the FASTQ format. So each sequence, each sequence is called a read, R-E-A-D, okay, read. These are short sequences, could just be 50 bases or 100 bases, something like that, okay, short sequences. So each of these reads are characterized by 4 lines. The first line is the identifier line, like greater than. You have become familiar with FASTA format. So the first line is greater than, and then the name of the sequence or organism and so on. Okay. That is the first line, identifier line. The second line is the sequence. Okay. And it's a DNA sequence, so it's a sequence of nucleotides. The third line is always plus, maybe for some reason it is always plus. The fourth line is indicating the base quality score, and it is coded. Okay. So you will have to decode it, and there is information about how to decode. Okay. If you just type FASTQ format, there is an information at different pages on how to decode this. So you can decode, and that will tell you the quality of the base that was sequenced. How good was it? Okay. So then you will have confidence that, okay, this base is good, the quality is good, if the quality is good. Okay. So 4 lines for each read. So typically there are often millions of reads. So you will have 4 multiplied by millions or whatsoever. That's why these files are really very, very big. So, and here is the formula for quality. So this is called pH rate quality. This is the score. So PE is the probability of incorrect base call. Okay. So if the probability of incorrect base call is 1 in 10, so there would be one mistake in 10. Okay. So then this would be 1 divided by 10, right? And 1 divided by 10 is 10 power minus 1. So log 10 base 10, 10 power minus 1 is minus 1. So minus 1 multiplied by minus 10 is no, minus 1 10. So this is the score, right? 10. Okay. For 1 in 100, the score is 20. 1 in 1000, the score is 30. Okay. So generally here, I think you would say that, okay, it would be good if the error is only 1 in 1000 or 1 in 10,000 or so, right? So, so now you understand the values of the quality score. Okay. What does 30 mean? What does 40 mean? And this is coming from this formula. Okay. Good. Okay. So I think, yeah, this is a display from a program FastQ that will help you in visualizing the quality of the data. So if they are lying in the green area, so here, so for each position, you have the quality score. So if the quality scores lie in the green area, it means good. If they ventured into red area, means bad. Okay, low quality. Okay, so here is the protocol and something that we discussed, that mRNA, and then mRNA is reverse transcribed to cDNA. So cDNA, and then cDNA is fragmented. So sonicate, there are methods for fragmentation, right? What are the methods? Sonication and what else? There could be many methods. Okay. So you may be knowing better. Those who do wet lab, they may be knowing of the techniques or technologies for fragmenting nucleic acids. Okay. So nucleic acids. So here RNA is fragmented. So there could be many fragments of RNA, typically of the same size, and then they are ligated with adapters. Okay. Also sometimes, because these adapters are identifiers, so you can do multiplexing. For example, you can have samples coming from different or individuals, and if you have ligated with adapters, you know which read is coming from which. Okay. So, and this is cost efficient. You can do sequencing at the same time of many different samples by this way, okay, by ligating with the adapter. Okay. And then once you have the RNA library, so this is the library or EST library, whatever you say, then you can sequence them. And each sequence would be 50 nucleotides or 100 nucleotides. So this is determined even before doing this experiment, that okay, what size you want, and it depends on technology also. Some technology can yield long reads. You may have heard about PacBio. Some can yield short reads, Illumina, you might have heard about Illumina. Okay, so once the reads are obtained, they can be mapped back onto the genome. Okay, so here it is shown that this is arising from some gene and it is mapped onto that gene. Okay, so and because this is messenger RNA, so these reads are coming from the coding regions, right? Not from intronic, from exonic. So mature messenger RNA. Okay, so so they are mapped onto the coding region. So you can see that this is intronic region. There is an intron here. So there are no reads that will map onto this. Okay, so these reads are mapping onto the coding sequence, and then you can assess the RNA expression level at each of the position. Okay, at some position there would be many many reads that would map or that would span that position. So which means that there is more expression level at that position, right? So this is what you can. So typically, actually, you can quantify the expression for a gene based on the counts of the reads mapping onto the gene. Okay, so if more reads are mapped onto the gene, then more is the expression level. Okay, so but when you map, then you have to be careful about some of the things. So it is possible. So let's look at two confounding scenarios. The first scenario is sequencing depth. Okay, sequencing depth. So your sequencing depth could be high. It means that you have sequenced so many times that you have covered the genome maybe 100 times. Okay. On the other hand, for some sample, you have covered only 10 times of that genome. So that's lower sequencing depth, right? So if it is so, then more reads could be arising because of higher sequencing depth, not because of these transcripts expressing more. Okay, so more reads are because of the higher sequencing depth. Okay, more sequencing depth, not because of the gene expressing more. Okay, okay. So you have to account for this. So basically, you have to normalize with respect to sequencing depth. The second scenario is length bias. So longer transcripts will have more reads mapping onto them. Longer transcripts will have more reads that will be generated from them. So when you will map back, you will have more reads mapping onto them compared to a short transcript, right? And this is because of the difference in the lengths. So that could also confound. Okay, so longer genes can have more reads than shorter genes, right? So this difference is not because of gene expression. This difference is because of the difference in the lengths of these two genes. So you would want to control for that, control for the length bias. So you would want to normalize with respect to gene lengths also. Okay. Okay. So here are the metrics which are the normalization for which has considered the normalization for sequencing depth and length bias. Okay. So the first one is RPKM. RPKM is for each gene or each transcript. Okay, reads per kilo base of transcript. Okay, so reads per kilo base of transcript. So you will divide by the length of the transcript and you will multiply by 1000. Okay, that would be reads per kilo base of transcript, and then per millions of reads mapped onto the genome. Okay, so you will divide by all the reads that mapped onto the genome and then multiply by 1 million. Okay, so it would be per millions of reads mapped onto the genome. So this way you have normalized with respect to both length of the transcript and the sequencing depth. Okay, so this is RPKM, and similar to RPKM is FPKM. So FPKM is analogous to RPKM. The only difference is RPKM has often been used for single end sequencing. FPKM is used for paired end sequencing. Okay, and more recently TPM, which is transcripts per million, that has been adopted by the users, TPM. So this is also a normalized metric, and a characteristic of this is that sum of all TPMs in samples is the same. Okay. So yeah, just to understand that you have to normalize, otherwise your comparison will not be fair. You only want to know whether a gene is differentially expressing or not, so you must control for other factors that could be contributing to the difference in the gene expression. So there are two main contributors to the bias. One is the transcript length and the other is sequencing depth. So you need to control for both, and metrics have been provided, RPKM, FPKM, TPM, that control for these two. Okay. So once you have the normalized counts, you can use the normalized counts for controlling for these. You can use the normalized counts for assessing the difference in gene expression between two conditions. Okay, that is called differential gene expression. Okay. Okay, so I think we discussed about this normalization of counts, and once you compare and you find that there is a difference, as I said that okay, there is a gene in me and I am sick that has normalized count 1000 and a healthy individual has 100. Okay, so there is 10-fold difference. Okay, 1000 by 100. So 10-fold, and this is called fold change. So fold change is equal to 10, and then you would want to assess the statistical significance of this. Is it really significant, or is it just by random chance? Okay, just due to some biological variability you are observing this. So that is statistical significance test that you would want to perform to assess whether the value of fold change is significant or not. Okay. Okay, so and you can see here in this table fold chain, and actually it is log fold chain. Log to help you visualize better the genes that are down regulated and those that are up regulated. Okay, so negative for down regulation, positive for up regulation. Right. So if fold change is 1, then nothing, right? No change. No change. So logarithm of 1 is 0. So that's why. So then the baseline is 0. Below that, down regulated, and above that, up regulated. Okay. So that's negative, down regulated, positive, up regulated. So I will go a little fast because we are running short of time. Okay. So fold change you understand, and significance of fold change. Biologists typically use both fold change and significance value, because sometimes the fold change could be very low, but still that value could be significant, and they are just not interested in those genes. They want to be getting a conservative set of genes that are differentially expressing, so they used more criteria for filtering. Okay, inferential statistics. So this is what is inferential. So hypothesis testing, statistical significance. And here is actually from a paper that we published in 2015. So you can see the reads that were mapped onto different genes. So can you see the reads that are mapped, that are shown in colors? Okay, so you can see that this is GNAT family protein, and in the control there are fewer reads mapping onto this gene. On the other hand, here is the plant actually subjected to highlight stress. Many transcripts were generated compared to control. So you can see so many transcripts whose reads mapped onto the gene. Okay, so is it upregulated or downregulated? Upregulated, right? Because relative to the control, you have many more transcripts that got generated. They are represented by the short reads that mapped onto the gene. Okay. Similarly, WRKI40 got upregulated, KMD1 got upregulated, and this JAT12 also got upregulated. Just to help you understand that, yeah, this experiment was done and RNA was collected within, I think, 10 seconds or so. So ultra fast changes that happened in the Arabidopsis plant. So yes.
Is we have the tutorial also, no?
Yeah.
So can we take up this one
Yeah.
in the post lunch?
Yeah.
Because post lunch session will be almost available.
Oh, okay.
Because this is needs some discussion.
Yeah, right,
and,
right,
and
right.
more techniques are there I can see.
Yes,
Yeah, so
right,
that will
right.
be better if you spend more time on this.
On this, right.
So I can
Then,
break
yeah.
at 12:30 is there. So
Okay.
we break from here.
Okay,
And then
okay.
I take the evaluation part.
Okay. Okay, right. I think I will finish in five minutes. Okay. Okay. So yeah, let's look at descriptive statistics, which is basically clustering. Clustering is grouping of objects. Okay. So this is something we already discussed, UPGMA. So if there are 5 objects, you can group them. For example, A and B are closest to each other, so they are grouped together as AB into one cluster, and then you will repeat this recursively, just like UPGMA. Okay. Then D and E will get grouped, and then C will group together with D and E, and then all will group together. Okay. So this is called agglomerative clustering, agglomerative hierarchical clustering. Okay. And the other is divisive clustering, where you are given n number of objects and you want to partition this full set of objects into 2, so that they are maximally different from each other. Okay. So divide into 2 so that they are maximally divergent from each other, and do it recursively. So when you will divide it, for example, then you will get 2 groups, CDA, CDE and AB. And then CDE will get divided into C and DE. So this is recursive again, and eventually all will get divided into individual objects. Okay. So both these process results in a tree. So one is agglomerative from left to right, and the other is divisive from right to left. Okay, and here is an example of uh, agglomerative hierarchical clustering of genes. So you can see through this method, the genes have got clustered based on the gene expression pattern. Okay, based on the abundance of the RNA transcripts. And at the top, you can cluster genes, or you can cluster samples also, or both. Okay. So at the top, you have the clustering of the samples. So, and I will skip this actually, and quickly in 2-3 minutes, I will show you the RNA-seq pipeline that you can implement at Galaxy. And this was provided to me by my PhD student Ahmad. We should thank him for providing these slides. So this is an experiment done with Bacillus and Thracis strain volume, and RNA-seq data were collected when this strain was subjected to oxidative stress, okay, hydrogen peroxide stress. So, for example, to understand RNA-seq pipeline, we got the samples for wild type. Actually, I can I do this during the tutorial, right? Because this is that. So
Thoda explanation.
yeah, this will, okay.
So we break racks now, okay? And now we get ready for the evaluation. So 1:30 time is there, because the lunch is at 1:30. So we take this 45 minutes for exam.
करते
ठीक करते जाएंगे, जल्दी हो जाएगा। हमको लग रहा है कि 10
मैंने
मिनट
45 मिनट दिया, वैसे 30 मिनट्स में कर
उससे
लेंगे।
पहले ही कर लेंगे। ओके ओके गुड।
सभी सब लोग हैं यहाँ पे, कोई मिसिंग तो नहीं है? एक बार
Okay.
देखो अटेंडेंस से तब
ओके
फिर-
वेरी गुड क्वेश्चंस, एवरीवन हैज पास्ड।
सब है, देखो कोई मिसिंग नहीं है। यू कैन ब्रेक द और लिंक चल रहा है ना? लिंक है ऑन कि नहीं? लाइव है ना? हाँ। So this evaluation is based on whatever you have learned till now, okay, and also on your general understanding. All are present? Okay, so you distribute it.
तो 10 मिनट में हो जाएगा।
Because it is MCQ, so take little time. Okay, don't do very fast. Okay, though it is 30 minutes time it will take. We have given 45 minutes. So you can use 45 minutes. There is no problem. If you want.
If you want,
Yes.
yeah.
Otherwise,
I am sure you all can do sooner, so you should be able to.
and if you are not able to understand any question, please don't talk each other. Because this is an exam. Please don't talk to each other. See only your question paper and do that.
Oh yeah.
Yes, यहाँ पे थोड़ा सा ब्रेक कर लो, चारों साथ मत
अच्छा यहाँ से कर दीजिए।
सब नहीं, यहाँ थोड़ा ब्रेक रखो, इधर आ जाओ, नहीं तो किसी का ऑप्शन गलत हो जाएगा। देख क्या क्या हाँ नहीं, वही मैं कह रहा हूँ, यू टोल्ड एवरीबॉडी इज प्रेजेंट।
Because she was present in the first lecture.
No, if it's not evaluation, she will not get certificate. Okay.
Do you have her phone number or?
उसको तुरंत बुला लो।
Not well.
Not well तो मैं क्या कर सकता हूँ?
No, but
See, don't talk these hypothetical things.